And welcome everybody to another BioSolveIT workshop. As mentioned already, my name is Alex and I will be your guiding light through the presentation Kinase Ligand Design, where we'll start with hinge binders and evolve them into full-fledged inhibitors. I love kinases, and kinases are a very popular drug target if you would check on the latest FDA approvals in the past decades. And from a drug discovery perspective, kinases are more or less very, very similar and also their binding site. Yet, every now and then we are facing a lot of challenges, mainly also due to the reasoning that the binding site is pretty well conserved within kinases, especially the hinge binding motif site or the hinge binding motif area. We still do face a lot of other problems like selectivity issues, as you mentioned already during the poll. And every time you are facing a drug discovery challenge, you end up with a point where you just struggle with your own kinase. It worked for every other kinase, and people are just reporting success stories here and there, but you're the only person that are now facing like a dead end. And this is what we are here for, where we learn a few new techniques, how to actually ideate compounds and get something useful out of your projects. The framework of this workshop is we'll talk a little bit about the design of kinase inhibitors itself, and we dive into two fields, not only the structure-based drug discovery, where we will work with protein itself, but we'll also look into a ligand-based project where no structure may be available, or you would just like to play around with more different techniques. As for the flow, I will start with an introduction. We'll talk a little bit about the ATP binding site, so the orthosteric binding site where the endogenous ligand binds, and then do a little bit about the hinge binding motifs available. And then we will dive into the structure-based drug discovery, where we'll do a lot of compound ideation, come up with new ideas, how to decorate a particular pharmacophore, also how to process your target itself and check for pitfalls or selectivity issues, differences in the binding site, and also talk a little bit about the property improvement of your ligand. And every now and then I will sprinkle some ligand-based drug discovery perspectives onto it. We will talk about the chemical space exploration a little bit, at least on the surface level, and then also talk about ligand alignment in 3D. So as I mentioned already, kinases are very popular targets, not only within the area of approved drugs, but they're also pretty popular targets for computational chemists and medicinal chemists. And maybe a controversial opinion, but kinases are actually pretty easy targets, or at least very accessible targets for structure-based drug discovery. And if you would look into the totality of all approved kinase inhibitors, there are actually reports that 25 of them, which is approximately 30%, were actually somehow involved in structure-based drug discovery. So one reason for that is that the kinases are comparatively easy to crystallize, so they have a very, very good informed idea about the binding site itself and how to approach this one. And also due to the improvement of methods within the computational area, we also have a lot of tools in our portfolio how to actually design drugs. And with computer-aided drug discovery, we can do a lot of things. One is to increase the selectivity. As kinase inhibitors tend to cover a lot of kinases, it's not that trivial to find something that is explicitly selective for your target of interest. And with also the perspective of the 3D arrangement of ligand and target, you can exploit a lot of interactions to either improve the potency or the selectivity profile. but also given the fact that we now have a lot of methods to ideate compounds, screen a lot of libraries, we can also do a lot of stuff in the FB area, so improve the properties from metabolism to physicochemical properties. And last but not least, what is also very interesting is that with the available methods, we also get a lot of tools to actually assess water molecules within the binding site to discover which ones are very conserved and therefore also to exploit those interactions within the drug discovery and drug design area. Now a little bit about the orthosteric binding site. In the kinases, we have the ATP binding site. So ATP is quite common or well known as an energy unit within cells. And if we look into how ATP binds in the binding site, we see that the nucleobase binds in a hinge binding site, and then we have the prolonged phosphate chains projecting towards a comparatively hydrophilic binding site, a binding area. A popular description of popular term that pops up when you talk about kinase ligand design is the hinge binding region. The hinge binding region consists of particularly 3 residues. You have one, pointer. You have the gatekeeping residue, and then 3 following residues here. Those are gatekeeping residues following the gatekeeper residue, namely 1, 2, and 3. And in this arrangement, you will see that almost all exclusive hinge binders actually interact also in this region by forming interactions with the backbone, not the side chains, with the backbone formed by those residues. Just to make it very complete and holistic, also for people new to the kinase area, a very popular motif close to the phosphate chains is the DFG motif, consisting of 3 residues that is comparatively very conserved among all kinases, and also pretty conserved domain is placed underneath the DFG motif, namely the HRD motif below. A little bit more into the perspective of the topology of the ATP binding site. This region responsible for the binding of the nucleobase is the hinge binding region. Then we have a comparatively exposed area where the sugar of the ATP binds. Some people also further differentiate into the front pocket and the solvent exposed pocket. And what's addressed by almost all kinase inhibitors is a hydrophobic back pocket close to this region that can be further subdivided into 2 areas, namely 1 and 2. And if you would take a look, now this kinase inhibitor is an example, you would notice that this region is not only exclusively addressed by very hydrophilic moieties, but also by quite lipophilic groups. And here you can see that there's actually quite a lot of space to actually exploit this lipophilic region to increase the potency and the selectivity of the compound. As for the chemical diversity of the compounds, you will notice that a lot of hinge binders are already reported, and from my perspective, they are also chemically diverse. So this is where you can play around with selectivity and also with the metabolism. So every one of them has different metabolism pathways. As for the solvent exposed pocket, they will have a lot of freedom to pick from. Due to the fact that this is a solvent exposed pocket, you could also obviously go with something very hydrophilic here to also increase the solubility of your compound. And last but not least, as I mentioned before, the hydrophobic pocket is quite large in some kinases, and therefore you have also a lot of freedom to play around with stuff in this area. This is something that I was posted on our social media accounts a while ago. The hinge binding motif set, it's a cheat sheet. I would just like to use this one to highlight actually that hinge binders don't have the one binding mode. It also can happen that if you introduce an amino group or amide group to the structure, that the arrangement within the hinge binding site will also change a little bit. Even compared to like shinolone and isochinolone, you will see that the arrangement of the lipophilic of the second ring system also may change. Quite important from my perspective, actually, is that one interaction must happen for all hinge binders, and this is the interaction with the gatekeeping residue 3 and this NH group here. All hinge binders do display this interaction motif here, whereas for the gatekeeper residue 1 and 2, the carbonyl here, it's not completely required. This is important to know. Throughout the industry, people still do visual screenings where they apply constraints on each side. Namely, this one is mandatory, and at least one of those must be fulfilled as well. This is the common standard that I have seen in our collaborations with the industry and also in publications where successes were reported. Yet the only one real interaction that all of them display is the H bond formed in this area, as you may see here and here and here as well. If you need this little cheat sheet, you can also get it from our website or follow us on LinkedIn, and then you can get access to it. It's a quite nice thing to put on top over your desktop to have it always on your mind and take a peek if you need it. Okay, so this was the introduction that we all are talking about the same things. I will start with the YAC1 kinase with the PGB code 6GGH. And also would like to mention, we have a pretty nice set available for download on the website, completely free of charge, of 44,000 hinge binding, no, it's a little bit less, a collection of 14,000 hinge binders as smiles, so you can use them for starting points for visual screening, for compound ideation. So you can download on our website, and if you need guiding lights where to get them, just post it in the chat and Justin will help you out. Okay, so now I promised you some fancy stuff, and this is what is about to happen. Let's dive into our drug design dashboard Caesar. So for my first molecule, what I will do is load the structure that I've mentioned, namely 6GGH, the human YAC1 kinase. Once I retrieve my structure, a lot of people were not familiar with our software. I won't go too much into detail on every single aspect because it would take too long. I just like to focus on the kinase design. In Caesar, it's required to select a ligand of interest, if one is available, to define the binding site. This is what I'm going to do now. So once I have my binding site defined, the residues forming my binding site, including also water molecules in the periphery, will be taken into account for further calculations. First, I would like to zoom in on my compound, so I select the compound here and use my space key to zoom in. And here we are, this is our first hinge binder. In this section, I would like to focus on the structure itself. So not an emphasis on the ligand, but more on the structure. For this, I will first hide the water molecule so we are not confused. Oops. And then I would like to know what structures share a same topology to my binding site. For this, I will load this structure in the protein editor. And then, by clicking on this button, I can access the Sienna server. The Sienna server retrieves structures containing sharing a similar binding site based on topology. And this is quite important. It's not sequence guided, so not the amino acid sequence, but it's guided by the topology. And what I get now are different structures that are already available, so compare similar PDBs, and I will load them into the protein mode. Again, I have to confirm a ligand here, and then I will take another one, so we have two additional structures here. There we are. So I will color them so we can differentiate them, one in a stylish pink, one in a vibrant green, and my original one in blue. Again, I will hide the water molecules so they don't bother us too much. And then what I will do, I will now realign them once again. And here we are. What happens here is if I now focus on the binding site only, I will see a sequence of the alignment of my residues that are now loaded here and are also overlaid in the binding site itself. What CSER offers to me is the possibility to highlight residues that are not similar. So if I now increase this similarity to 2 angstrom, similar residues will disappear, and only those with high this similarity will appear. What do I mean by this? So if I now focus on this glutamic acid here, I can zoom in. You will notice that those residues actually flip around. This gives me a lot of ideas about what residues are highly flexible. Same goes also for this arginine. And now if I hover over this arginine, I will see that this arginine does flip a lot, which I should also consider during my docking procedures and also assess for interaction potentials. So maybe I wouldn't trust interactions that are formed with this arginine when I perform rigid docking. Now let's load something else. I will load another side type of the kinase. And again, align it with my previous structures. And color my new kinase in yellow. So this is the YAG2 kinase. What happens here again, if I click on my button, again, these similar structures will be highlighted. This also helps me to see which residues are not conserved among the subclasses. Like in this case, for the YAG1, a phenylalanine is present here, whereas for the YAG2, I have a tyrosine here. And again, I get a lot of ideas. So our previously investigated glutamic acid is replaced by an aspartic acid in this area. So this is also something where you can see some differences between binding sites that you can address during your compound ideation process and also to increase the selectivity. Okay, so much for the structure assessment. From my perspective, AlphaFold is also something that you can use. The AlphaFold models for kinases are very, very strong because there is a lot of information and data available on them. So they also represent pretty legit points, starting points to be used for your design campaigns. So for my next miracle, I will remove the previously loaded structures because I don't need them any longer and dive into the docking mode. So I would like really to have the very core of how you would approach things. So let's start from scratch. The first thing I will do is I will load some structures as hinge binders. For this, I have prepared a small set consisting of SMILE strings and only 20 molecules so far. So this is where you can see them. They all come from the hinge binding set that we have created. It's also available on our website. So to give you a little bit more of perspective how this hinge binding set was created, we took the whole Campbell database and extracted bioactive compounds displaying activity less than 1 micromolar, and then cut those bioactive molecules into smaller fractions, so that only the ring systems persist. And then we decorated them additionally with amino and amido groups just to reflect the functionalities present in known hinge binders. And what we did then, we docked this whole generated set of, I don't know, 20-something thousand on different BDB structures, and only those that were survived and were able to bind within the hinge binding region ended up in this binding set. And this is just an Excel for them, so only 20 compounds. Now to the docking. And CSER, it can always be a good idea to actually perform docking in the hinge binding region. I have to see the hinge binding region, and there she is, namely this area in here. And for this, I would like to do a pharmacophore constraint docking. So first things first, I will select this very important part. This is where I've mentioned that this residue should obviously interact with the hinge binder and will display the perfect spot for an interaction contact. I did this by clicking on this one and then show possible interactions. Now I will place my constraint on this area and make it comparatively small because I really would like to have an acceptor in this area. The next thing I will do is I will define 2 additional interaction spots because this is how the industry handles stuff. I will select this carbonyl group of the GK+3 residue, define a new constraint. Again, I'm displaying the perfect angles for an hydrogen bond interaction and place a constraint in this area. Let's stick with 1.5 angstrom for the radius and select an donator in this area. Next, what I would like to do, I would like to place a constraint on JK+1. Again, define constraint, show possible interactions, place it here, and let's go with 1.2 angstrom. And this is the perfect interaction spot for a donator again. Apply. So for me, it's quite important that this nitrogen is satisfied. So I will keep this one as non-optional and mandatory. As for the other 2, it's fine if one of them is fulfilled. Therefore, I will make both of them possible, and at least one of them must be fulfilled. That's it. Now I will just start the docking by clicking on the docking button, and within a few seconds, we will end up with the results. By default, Caesar generates 10 docking poses per ligand. If you would like to go higher, 500 is the maximum of poses that you can generate within Caesar. So I'm done, and I have now 200 poses. And as we can see, they do what they are supposed to do. Since a lot of you are not familiar with our software, those green spheres around an atom are our so-called height spheres. They give us an idea about how satisfied an atom is within the binding site. Green means it is satisfied, red means there is room for improvement. So to fully assess my docking results, I will select all of my generated poses and transfer them to the analyzer mode. The analyzer mode allows me to group my molecules. So basically, if I open up my filter and activate the group molecules, it will filter my molecules and only display the best results to me. So out of all poses, 20 poses per ligand, only the one with the highest estimated affinity from our side will be presented. I will now rank my compounds, and what you will also receive are actually color indications in regards to the LLE, torsions, and clashes, so you can easily assess your compound complex and decide if you would like to work it with or not. And those are the results for the initial fragment binding spots. And as you can see, they all display at least 2 interactions, which makes me happy and should make you happy as well. And there we go. Oh, okay. So much for the docking. Now let's get into the compound ideation process. What you can do with Caesar is to actually play around with your compound in the old-fashioned way. So you can load your compound in the so-called molecule editor mode, and then just start decorating. So if I now click on this hydrogen and push the C hotkey, I will add a carbon in this area. Once I'm done with the editing, I can export my molecule and then receive an assessment of if the affinity has improved or not. And it seems like that the meta group there is pretty happy. What you can always do, you can also activate the binding site representation. So I'm going to do that now to get also a feeling for the depth within the binding site and also to assess where area is available to grow into. And here we are. This is the binding site. I can also make it transparent. And another nice feature to assess where room is available for decorations, if you go into this visualization settings, you can also turn on the unoccupied space. What the unoccupied space does, I'll quickly turn off again the binding site, it introduces those foggy spheres, and those foggy spheres are an indicator where one fluorine atom has enough space to be put into. So basically, you see where there are still unoccupied areas around your compound. Let's turn it off again and then activate our surface. And this is how you would operate in the old-fashioned way. Obviously, you can go wild. So introduce a flow line there and export it and see if anything happens. Again, doesn't feel a huge improvement. You can also remove stuff. You can introduce additional ring systems, maybe add cyclopropyl. Let's see. Yeah, it's not completely happy, but a little gain in affinity is there. So this is the old-fashioned way, but we are not old-fashioned, right? You can use the molecule to kind of like play around with hypotheses when you are in your group and you think about if it doesn't make sense to introduce nitrogen in a ring system or not. But if you really would like to explore the whole proximal chemical space around a compound, we have better tools. For this, I will now take my initial compound and load it into the inspirator mode. See, this inspirator mode is the mode that allows us to browse through a lot of ideas to come up with improved, better compounds. The first thing I would like to show you is the analoguing button. By default, the analoguing button creates 10 different solutions. But again, you can go up to 500, but to keep the time frame a little bit shorter, let's stick with the 10 for the moment. If I push the analoguing button, it will perform over 100 medicinal chemistry transformations on my ligand to check which common medicinal chemist decorations or bioisosteres can improve the affinity of the compound. So it will check the area, the surface area, the volume, the binding site itself, and take it into account and present to you only stuff that is actually, not doable, but legit. Let's call it like this. It would never introduce a meta group that would lead to a clash. So it takes all of those things into consideration to only show you the compounds that would be at least would make sense to follow up on. But it would not show you something that is kind of like just a full enumeration of all possibilities, because it would just waste computational resources. And it also here it communicates with you, I'll get a little bit larger, what transformation was applied here and acetylene was introduced in this position. And if you check now all compounds and make them visible, you will also see where enough space is available for going strategies. So this was the analoguing mode. Quite interesting for medicinal chemists to just kind of like think outside the box, because medicinal chemists tend to do the same thing over and over again. And this is where new things may come into motion, and they will think, oh yeah, I didn't consider to put a G-fluoromethyl into this position. Okay, let's get rid of this stuff and now dive into the good stuff. So by starting with my compound here, I would like to show you a little bit of the fast grow tool that is implemented into Caesar. For this, I need to select a growing point. Let's take a look here maybe and go from this position, because the space is there. I mean, why not? And now I will go into this section and increase my number maybe to 20. And as you will see, once I actually add my constraint here and define this is the exit vector I want to grow from, fast grow becomes active. I will now click on it, and as a background, fast grow screens a fragment library for decorations that can satisfy this binding site. What is very important about this one, it's incredibly fast. Right now, I'm screening 120,000 fragments. 120,000 fragments with 10 conformations of each. So basically 1.2 million conformations within seconds. You saw it live, it's not like pre-recorded, otherwise I wouldn't have issues in the previous setup. So it's incredibly fast, and it provides you with a lot of ideas how to decorate your compound. As you can see also, the affinity was also assessed directly, and I can see improvements every now and then. And this is what comes up. Basically just projecting into the binding site. And the idea behind FastGrow is it tries to complement the binding site shape-based. So we project into the binding site and check for the available space. We take also into account the possible hydrogen bond interactions and lipophilic interactions. And this is what comes out of it. An amide here, which also forms an interaction in this area. Personally, I really like the results. It's no surprise, we can also see here that those ring systems are actually pretty well accepted in this area. So therefore, it's kind of like a perfect match for this part. And you can see also the diversity of the results is quite high. And I really like this one. So it looks very metchamish to me, at least. So maybe let's follow up with this one. What I'd like to do now is actually to show you a little bit about the chemical space exploration. But before we do that, I would like to start one additional one within the docking, which will take some minutes. And before we will sit in front of each other in silence, I would like to avoid that. So we'll now clean up a little bit and load some molecules for the docking to be run in the background. Let's go with them. And there they are. So just for you, if you would like to perform a template-based docking, so I really like this compound. I will turn off the pharmacophore constraints here. I will select this one as a template and dock the other 100 compounds in the meantime by pressing on the template-based docking, which applies a maximum common substructure method. So basically all compounds will be aligned to my template based on the maximum, the largest common substructure that they share with them. Before we dive into the chemical space exploration, we offer 3 different methods within our second platform, Infinity, that allow you to screen for different compounds, because similarity is something different depending on the questions you ask and also the compound that you use. And therefore, different methods are required to also address different aspects of the drug discovery process. One algorithm that we offer is called F-trees and is hidden as the scaffold hopper within Infinity. And you can use the scaffold hopper to screen for similar compounds based on their pharmacophore features. It's quite fuzzy, so it has a lot of potential to find novel intellectual property and novel scaffolds. And I really like it. And it's very different to other comparison methods like fingerprint-based applications. The second algorithm that we offer is called SpaceLight. The mode for this one is called Analog Hunter in Infinity, and it screens for close analogs with high Tanimoto similarity. So basically, the fingerprint in Infinity is the ECD SFP4, quite common, and this is the golden standard for the industry, I would say. This is also where the CompCams are the most familiar ones with, so they know what to expect, and it's quite nice also to use for the exploration of the proximal chemical space around the compound. Last but not least is SpaceMax, the third algorithm that we offer, and this is a substructure matching method. So based on maximum common substructure, you retrieve compounds of interest, whereas the rest of the molecule is ignored. Very, very interesting for fragment-based approaches if you would really like to have a set of, let's call, 1 million compounds that contain one particular hinge binder that you're interested in. This is a method that you could apply to extract those 1 million compounds from the chemical space. What's very interesting also that the chemical spaces come in different flavors. As of now, we have 7 different chemical spaces available. And now to compare just 2 results, this is the query compound that we have extracted from the inspirator mode. And if you would ask for this compound with SpaceMax, this is what you would retrieve from the space. And there you go. And as you can see, there's a minor difference. You get a ketone here. So this one is no longer an amide, but an amide, therefore neutrally charged. But you get a lot of decorations in this area, as you can see here, also in a minor group here. If you would use the same compound in the real space, you get, I would say, more closely related compounds. So we still have this functionality here with a positive charge. And you also get a lot of stuff in this area that is different. So basically, a morpholine ring, which could increase the solubility of your compound, and also different decorations in this area. So therefore, my recommendation in this area is always to screen several chemical spaces if you have a compound of this, just to get a feeling for what they can offer to you for one compound. And the best part is actually, obviously, for those also who don't know, everything that you retrieve from our partners from our partner spaces is also commercially available. Especially the guys from Enamine who created the real space. If you would order those 6 compounds, they are quite, I would say, cost effective in this regard, very competitive pricing. And you would receive those compounds within 4 to 6 weeks to your table. So they synthesize this stuff on demand, and it really, really accelerates the whole drug discovery perspective. So therefore, I would like to show you how to operate in Affinity. And for this, let's dive into the analyzer mode. So Affinity also contains an analyzer mode. And I will now load a few compounds here. The first compound I will load is actually the results from the real space. So again, this was a SpaceMax search based on the maximum common substructure. And also, let's load the Kinases results. So what I have now is a set of 200 molecules. The analyzer mode allows you to process your compounds, analyze the data set. There's a lot of things that you can do, like creating histograms. Let's create something for molecular weight. I click here, histograms, and then here for molecular weight. And I see distribution of the compounds that are inside my set. I can also create the same stuff for log p values and anything else that is numerical, which you can see here. Besides, from that, I can also cluster my compounds based on a Burmese-Merkel scaffold, which I find very interesting. So you can also use this to dive into particular Burmese-Merkel scaffolds to select one from each set to increase the chemical diversity of your compound selection. You can also cluster the Burmese-Merkel skeletons if you're that kind of person. And what I really, really like is also the possibility to actually fine-tune your results. So I'm not a huge fan of halogens within my compounds. So to just have kind of like the naked molecule, now filter and remove all halogens in this set by putting it in one. And then there we go. 96 compounds survive the process, and also my scaffolds will reduce in this area. Okay, now, how do you operate within the chemical spaces? The easiest thing is actually to load your compound. You always need a compound to operate within CSAR. I will now throw this one into the analog hunter to retrieve very close analogs of my compound. Now I have to select a chemical space I would like to search in. Let's go with the real space. And just to have everybody on the same page, those are 48 billion compounds. Okay, keep that in mind, 48 billion. And now I will click on my start button. The default settings is I will retrieve 100 compounds. So I'm browsing now through 48 billion compounds live in front of you. And this is the time that it takes to actually process everything. You can watch on your arm wristwatch. It should take more than a few seconds. And here we are, 48 billion screened for the closest analogs based on ECSFP4 fingerprint presented to me. And you see the results are also highlighted here. So I can also see the similarity between my compound and the results that I'm now presented with. So for structure-activity relationship analysis, this is the go-to mode, because this is where you can explore what is available commercially, and you see the differences here are what you would actually pick for those kind of analysis. The other mode that is available right now is the scaffold hopper. So I will just use my initial compound, throw it in, and then search within the Kinases space. There we go. And this is a scaffold hopper. So this is where I retrieve novel scaffolds, novel ideas for pharmacophores. And I will adjust my parameters a little bit, so you have a little bit more of freedom here. What the target similarity should be, what's the lowest similarity you allow within your search. And also by playing with this button, you can increase the chemical diversity of your compounds. So with the total diversity at 1, you will have a set of pretty familiar compounds. But if you reduce the number here, you will get a more diverse set from the chemical perspective. Let's go with 0.97. Click on the Start button. And now it will take a few seconds. It won't be too long though. Meanwhile, as I mentioned, SpaceMax, the third search method, will become available within Infinity 6 in the next weeks. So keep your eyes open and your ears, so don't miss out on that. There you go. So we've screened 12 billion, and this is what we receive as results. Again, the similarities or the compared fractions of the compound are highlighted in the same color, so we can easily assess the similarity between both of them. And this is where you can ideate for new ideas for compounds and new pharmacophores. So again, this is what I always say, there is no one similarity. Similarity is always depending on the project and also on your target. And if you would ask 5 different people about their opinion what a similar compound is, you would receive approximately 7 opinions. So therefore, you need different search methods to actually operate within the chemical spaces and retrieve your compounds of interest. Okay, so this was Infinity, pretty straightforward. If you would like to follow up on those compounds, you can export them in different formats, smiles. You can also export them as SD file. So everything is can also be exported from Infinity to other platforms just to explore or apply your methods on the compound sets. Meanwhile, our template docking has finished. So this is actually my workflow of the compounds. So if I retrieve something from Infinity, and if there's a target structure available, I obviously always redock my compounds. And since I have a pretty decent idea about how my one compound binds, I use this one as a template, and this is why we did the template docking or started template docking ago. And these are the results. As you can see here, I can also highlight my initial compound in another color, maybe something like this. And then you would see how my other compounds are actually aligned or placed on my previous compound. And again, I can also score them like this. And this would be the most potent compound based on my docking predictions. And we are now penetrating the nanomolar affinity range. Okay, what's next on my list? A little bit more about the inspirator mode. So the inspirator mode is a little bit agnostic towards synthesizability. Yet, this is a workshop also for advanced users or for advanced usage. What you can do, you can actually guide your searches. So as I mentioned before, we had a lot of amide groups here. And if you would like to focus on particular features, you can also apply pharmacophore constraints within the inspirator mode. So for this one, I will remove everything but my initial compound. Reload it into the inspirator mode. And then I can apply pharmacophore constraints. So in this area, I had, maybe I have to redo it, unfortunately. I was too fast. Just the rotation point. So there we are. I will use this amide actually as an inspiration. So I will load this one into the inspirator mode, zoom in a little bit, and I will now define 2 new constraints. One here 0.8, and I will define it as an oxygen. And for the next one, I will place it here and define it as a nitrogen. I will remove now everything else and keep on going with the inspirator. So now my constraints are defined, they are still there. And now if I click on fast grow, 20 results, I will receive only fast grow fragments that also satisfy my pharma constraints. So basically an amide. And there we go, again, we screened 1.2 million, and the results are all containing an amide in this section. And this is how you can actually introduce some sort of synthesizability, because if you say, I can introduce an amide, I have the building block available, and it's just kind of like, what's the best building block to place in this area? Because you can synthesize this amide, this is how I would approach it, to introduce synthesizability into your results. Okay, what's next? Another thing that I have created exclusively for you is actually, let's go with this compound. In the next few days, I will also upload a new FASCO library to our website. I will just remove all constraints here. Oh, no, I did want to remove this one. I will remove this one. I will apply 2 constraints for the hinge binding region. And now I will replace this part of the molecule. Let's remove the old stuff. And select a new library containing only hinge binders. So this library contains 44,000 potential hinge binders. And again, I can just press on fast scroll. And they stem from the hinge binding set, so all are legit potential hinge binders. And we'll screen for candidates that can be placed in this region to satisfy the hinge binding interactions. Again, it can make sense to introduce pharmacophore constraints to guide your search in this area. Okay, maybe something else in regards to Recall, because not everybody is familiar with Caesar. So I will use this one as an example. Load it into the inspirator and delete the old stuff. As you may have noticed, once I add a constraint in regards to define an exit vector, one is placed here, and the part that we will place is grayed out. I can also do it like this. I can also place 2 constraints, and this is where Recall becomes active. What Recall does, Recall actually screens for 3D fragments that satisfies those both exit vectors. I define now one exit vector here and one there, and press on Recall to find fragments that can match this area. And here are my results. And you will see, this was my initial compound, a lot of flexibility, a lot of rotatable bonds that highly affect the permeability or cell permeability of a compound. And by using Recall, I can actually screen for compounds or for substitutions that are more static. So again, I'm using the PDB set, so those fragments are actually taken from the PDB itself, where we cut off every single 3D ligand and extracted those beautiful parts here. And yeah, those are the 3D arrangements that could satisfy us. One may argue, well, it's still highly flexible. No problem. We can also, again, apply pharmacophore constraints. So what I will do now, I will define a new pharmacophore constraint, placing it in the middle of those both things, and ask that any aliphatic chain here has to be excluded. There we go. I will remove my old results, and again Recall. And we also, again, here we're talking about millions of fragments that we are screening. Again, very fast. And I'm receiving a lot of very, or at least more static results in this area. So a beautiful tool to actually play around by reducing the degrees of freedom by removing rotatable bonds in this area. Okay, you can also use recall to merge fragments, but I think it would be a little bit too much for this workshop. So therefore, if there's any request for me to show it to you afterwards, please tell us in the comment section that I can follow up on that. Okay, we are reaching the last part, namely the ligand-based drug discovery in 3D this time. So I will now dive into the similarity scanner. For my first miracle, I will load now the data set for ligand-based drug discovery. And what we have here is a template molecule. You may recognize it already by now. And also sets of other molecules. So they can be anything. This can be from enumerated data sets, whatever. And my question is actually, can I find something that is very similar to my template compound if I don't have the structure or no idea what my target is actually? Can also happen every now and then. What I will do now, I will select my template molecule, and by pressing the similarity scanner 1 button, I will align my other molecule shape-based on my initial template. This will take a few seconds, again, very fast. And there we go. What we receive is again a numerical value that can be also exported. And if I rank my compounds, I can actually go higher. Again, I will highlight this one as purple. And those are my aligned molecules. What FlexS does, so FlexS is the algorithm behind the similarity scanner, it tries to match not only, it does a shape-based alignment. This is quite important. So it tries to satisfy the needs of hydrogen bond donors and acceptors, the shape and also the lipophilicity of a compound to actually match my query compound. And this is where the results come from. And this is also a virtual, you could use this one for ligand-based virtual screening. So if you really don't know the structure, if you don't know the target, load a ligand and then align your ligand based on that. So this is the baseline for the ligand-based drug discovery. Now an advanced technique. I will clear my similarity scanner, throw every molecule out. And for my next miracle, I will add a similarity scanner decorations because I really like this approach. What I'm doing here is actually ideating the decoration of my hinge binder. So as you may recall, this was actually the R group here was actually replaced by the hinge binding motif in my original compound. And this area here is just a decoration of my hinge binder. So what I can do as well, I can actually use my decoration and do a ligand-based alignment of the decoration only, without excluding the hinge binder. So an ideation of what compounds or what building blocks may be similar to my compound of interest. As you can see, the rest is pretty default molecules. And what I will do now, I will again select this one as a template and start my similarity one. This shouldn't take too much time. Few seconds. And there we go. So it tells me that 6 molecules were not superposed because there were just 2 different. Again, I will rank my results, display my original compound in purple, pink, and there we go. So I can also use this one, and you actually can see here how FlexX tries to accommodate smaller modifications to still match the theme, So we have the tyrosine here, it's flipped, yet it's flipped because it also tries to match the methyl group into the swing system. So ultimately, I would still consider this one to be very a good molecular match. And this is where I now receive or can see which decorations are also similar to my side chain of the ligand. Okay, one last thing before I finish my presentation. I'll clear the similarity scanner once again. And now load the smiles. So our previous set to have a full circle. What I would like to do now is actually to show you how you can perform ligand-based drug discovery also for hinge binders. So what I will do, I will select one hinge binder here, namely this little fella, and I will use a pharmacophore constraint. So by clicking here and show possible interactions and placing the ligand sphere to this area, I can also select an acceptor interaction contact. So what I'm doing here is basically I form a pseudo receptor pocket. So I don't know what thing is there, but what I'm telling the system is every ligand that will be now superposed on my template compound should also form or should be able to form an interaction with an NH or any form of hydrogen bond donor in this area. I apply my constraints, select it as an, was an 8. This one, select this one as a template and start my docking one. And there we go. As you can see, now again, you can also rank those compounds by similarity, but all my beautiful stuff is now aligned in that manner that it can also form an hydrogen bond interaction in this area. If you would ask yourself, why was this one not aligned? Well, again, using the original one as a template molecule and putting a focus on this compound, you will actually see that it will be very difficult for HB30 to be aligned because the overlap with my template molecule is not enough, and therefore the similarity score would be too low. You can adjust similarity score though in the settings by going lower. You will also see result for this compound. Okay, I'm done on my side, and this is where I would like to hand over to Justin.